Hello everyone, my name is Nilan Tan, and I will be presenting a report on the progress of my NSci 400 capstone, an exploratory research project titled Compensatory Mechanisms of the Rubrospinal Tract in Forelimb Motor Control. Before we can understand the rationale behind researching the specific role of the rubrospinal tract in spinal cord injury, we need to consider why researching spinal cord injury is even important. Surprisingly, spinal cord injury is all around us, as 250 to 500,000 people around the world are living with spinal cord injury right now. On top of that, due to increasing longevity, spinal cord injury is becoming even more prevalent as older people are far more susceptible to it. The consequences of spinal cord injury are even more severe. People with spinal cord injury are 2 to 5 times more likely to die prematurely, and they will suffer more while they live as well. They face a monetary burden of about 3 million Canadian in lifetime spending for hospitalizations, rehabilitation, physical therapy, lost wages, and more. On top of this cost, people with spinal cord injury face severe reductions in quality of life in the form of lost employment, interference with education, mobility issues, and social exclusion through stigma. So we've established that the spinal cord is significant as there are severe consequences when injured. But why look at the rubrospinal tract in particular when there are other descending tracts? Well, the rubrospinal tract is responsible for muscle tone, posture control, and motor control of limbs in non-human primates and other animals. This is going to be very relevant when we discuss the animal model later. The rubrospinal tract originates in the red nucleus, crosses the midline in the caudal midbrain, and terminates in the contralateral spinal cord, as shown in the diagram on the right here. Finally, and perhaps the most important reason why researching the rubrospinal tract is crucial is due to its plastic nature. When the cortical spinal tract is damaged, the rubrospinal tract has been shown to partially compensate the lost motor function. This finding opens new possibilities for neural repair post-spinal cord injury through treatments targeted at the rubrospinal tract. To prove this point, the Han et al. Paper explores the compensatory behavior of the rubrospinal tract using the SULSAR-3 EMX1 rat model, which blocks the formation of the cortical spinal tract. In panel G and H on the top right, the authors stain the axons of the rubrospinal tract and image the spinal cord. We see there's an increase in the number of projections to the spinal cord in the SULSAR-3 EMX1 rats visually, but also statistically counted out in panel I. In the figure below, the authors investigate the effects of the cortical spinal tract absence behaviorally in rats. Panel A and B show an open field test where rats are free roaming. The SULSAR-3 EMX1 rats actually walk more than the control, but the key takeaway here is that the experimental rats can walk relatively normal without the cortical spinal tract, which suggests a compensatory system in place. The bottom panel uses the serial handling test to show skilled behaviors. The authors score the animal based on forelimb angle, digit movement, and paw control. In panel J, the SULSAR-3 EMX1 rats perform significantly worse, suggesting that there's minimal compensation for skilled behaviors in the absence of the cortical spinal tract. While the Han et al. Paper gave us an in-depth understanding of the visible compensatory behavior of the rubrospinal tract, the literature still has many knowledge gaps and unanswered questions. Here, I'll point out a few. Firstly, what are the compensatory effects of the rubrospinal tract after spinal cord injury? The previous figures showed us about the compensatory responses during the absence of cortical spinal tract. But in real life, that isn't really how it works. Rather, we're far more interested in the compensatory effects after spinal cord injury. Secondly, what are the compensatory effects of the rubrospinal tract solely? The previous figures showed us rat motor control in the absence of cortical spinal tract, but there's no way to prove that it was only the rubrospinal tract that was compensating, rather than the other descending motor tracts. And finally, what are the molecular mechanisms behind the compensatory role of the rubrospinal tract after spinal cord injury? As this question allows us to approach the crux of the issue and develop novel treatments for spinal cord injury. All these questions led our team to develop this project's aim, to determine the molecular mechanisms of the rubrospinal tract compensatory behaviors after spinal cord injury in rats. So how can it be done? Let's discuss the gross experimental plan first. Using a cohort of 12 Long Evans rats, famed breed for their intelligence when it comes to skilled behaviors, they'll be trained for 8 weeks to excel in an array of tasks, which will be explained in an upcoming slide. A unilateral spinal cord injury in the pyramids, or a pyramidotomy, shown on the right image here, is used to completely sever the cortical spinal tract and rubrospinal tract on one side of the body. A DREAD AAV injection surgery, which will be explained better later, is necessary to allow for selective drug-induced silencing of the remaining contralateral rubrospinal tract. Two groups of rats will be formed where one group receives the silencing drug while the other one receives the saline control. After sufficient recovery post both surgeries, measured behavioral testing will occur before euthanasia of the animals. Finally, tissue will be stained for axonal and neuronal markers and data will be analyzed. For the behavioral testing and training, rats undergo 4 different tasks. First, the serial handling test, which we've previously seen in the background part, evaluates a rat's fine motor control through analysis of the arms, wrists, and digits. Observers qualitatively score the results according to the IBB scale, the metric for measuring serial handling, as shown on the top right image. Second, the open field test shown by the previous paper is also used as it measures gait, distance traveled, and regularity of paw placements to show overall motor control. Third, the food pellet reach task examines forelimb motor control over the ability to grasp food pellets as shown in the bottom left image. It is a quantitative metric as the rats are scored by number of successful grasps within a 10-minute period. Finally, the irregular ladder rung test measures the rodent gait, paw placement, and footwork recovery in an irregular terrain as the rungs are spaced out oddly as shown as the bottom right image. To understand the role of DREAD AAV injections, I need to explain the mechanism first. DREAD stands for Designer Receptors Exclusively Activated by Designer Drugs, and these receptors use HM4, which are inhibitory and silence the cells that the DREADs are expressed on. The DREADs are paired with an adeno-associated vector, or AAV, which is what allows the receptors to be expressed within the red nucleus cells. Through this system, the rats can maintain their spared rubrospinal tract's compensatory behavior outside of times when the drug is administered. For our experiment, we alternate between treatments of compound 21, the agonist for DREADs, or saline as a control when we conduct behavioral testing. The control group of 4 animals receives saline only in case there are any long-term effects of compound 21 administration. So what has been done so far? The behavioral training, pyramidotomy surgery, DREAD AAV injection surgery, and behavioral testing have all been completed. At this point, only the tissue staining and data analysis for both the behavioral testing and tissue staining need to be conducted. On the right are pictures of my rats during serial handling and ladder task. So what's next? I need to finish cutting all the tissue on the cryostat that's been collected from the rats, as well as finish the data analysis on the behavioral testing videos from the 4 different tasks. After this, I need to stain the midbrain and spinal cord sections from the rats using immunohistochemistry, where I'll stain for an axonal marker, a neuronal marker, as well as an amplifying antibody for the DREAD AAV. Finally, I will compile these results into a research report that will be submitted to the NSI 400 program, as well as prepare a poster presentation for the ICORD Research Day as requested by my supervisor.